You guys have the seminar in the morning, 9 o'clock in the morning? Every seminar?
Yes.
Right? And it's well attended. I'm very impressed. I'm very, very impressed.
I think we're ready to start. You guys come forward.
Let them have the rest of the donuts. Are they donuts, guys, there, or bagels? Donuts? All right. You need the sugar in the morning, okay? Trust me. Necessary so that you do not go to sleep. Have a seat, guys. All right. Well, let's start bright and early. I'm very, very, very incredibly impressed that at 9 o'clock in the morning, we have plenty of graduate students sitting here. So I will remind my own graduate students, I got 15 of them. It's very hard for me to get them at 9 o'clock. So I will use you as an example, and trust me. And so please do, in case they ask you, Do you guys really come at 9 o'clock? You will say, absolutely, we do every day, okay? So do not say anything else, all right? Well, good morning. We have Professor Rita Alevriadou, and she is an Empire Innovation Professor from University of Buffalo. It's quite cold up there, but not yet at this time. I think much better up there than what it is likely down here the last few days. We're happy to have you. Okay, Rita, we're happy to have you. I want to say that a little bit about her history. But before I do that, she is an expert in cardiovascular bioengineering, endothelial cell mechanotransduction, mitochondrial free radical biology. And that's a lot. I only understand it because I'm Greek, and that's the only reason I understand the words. But I'm sure you do. I'm sure you do. An incredible individual. I've watched her career, you'll see that in a moment, for many, many years, for at least, I would say, more than 20 years, almost 20 years plus. She's a graduate from a university called the Aristotelian University of Thessaloniki in Greece. I know that university very, very well. My wife is from near there. And it's an excellent university. And after 1986, when she graduated, she came, here to the United States, and she got, her PhD. And her PhD, after her PhD at Rice University, right? At Rice, is that correct? Yeah. After her PhD, she went and did postdocs at the Scripps Institute in La Jolla, California. That's a wonderful, wonderful institute. I wish I studied there myself. And also then became an assistant professor at Johns Hopkins University, not Johns Hopkins, Johns Hopkins University. Just that's something that everybody makes a mistake about. And that was an incredible also place and career. And then I happened to meet her at Ohio State in 2003 when I came from Michigan down there. So I just noticed that you came down at the same time. I came January 2003, and she came down also at the time. Yeah, a little bit later maybe. That's where I met her. She's an incredible scientist, a very dedicated person, and I can imagine that she will keep you alert for the entire 45 minutes that it's going to be. So without much ado, I will let her to come up and introduce you, Professor Alevriadou, to give you, a very nice presentation that I already see over here. So, please, Rita. Yeah.
Thank you very much, John, for the wonderful introduction. Very happy to be at FIU among so many friends and colleagues. I appreciate you inviting me to present my research. The title of my talk is Mitochondrial Transfer via Endothelial-Derived Extracellular Vesicles in Cardiovascular Disease. My laboratory, the Alevriadou Vascular Mechanobiology Laboratory, AVML, conducts research on vascular cell responses to changes in the cell's mechanochemical environment. We are trying to understand how these changes affect the cells and contribute to initiation and progression of cardiovascular disease. Our primary goal is to elucidate the molecular mechanisms that lead to vascular endothelial cell dysfunction. Why is this important? Because we know that all of these conditions, hypertension, dyslipidemia, hyperglycemia, basically diabetes, smoking, and aging, all of these disrupt the balance between free radicals, also called reactive oxygen species. The initial reactive oxygen species is superoxide radical, and then it converts to other types of radicals. And nitric oxide, a potent vasodilator that is released by the vascular endothelial cells in response to mechanical forces such as shear stress that is exerted by the flowing blood on the surface of the endothelial cells. And of course, the endothelial cells are the cells that cover the inner surfaces of all blood vessels. So they are continuously exposed to mechanical forces, such as shearing forces due to the flowing blood, and also as the vessels dilate and constrict, there are also other types of forces, such as stretch, and of course the normal pressure, the normal force on the surface of the endothelial cells due to the blood pressure. So when the cells are functional and healthy, the concentrations of nitric oxide are about 1,000 times the concentration of reactive oxygen species. So this balance, when it starts tipping over in favor of reactive oxygen species, then we see that the cells are facing a situation of oxidative stress. So what is oxidative stress? Is a state where the cell either overproduces free radicals or it produces a normal maybe range of free radicals, but it doesn't have the capacity through antioxidant enzymes to scavenge these free radicals. So there is a surplus of free radicals. And why do we worry about an overproduction of free radicals? Basically, when there is a surplus of free radicals. Because these compounds, the reactive oxygen species of free radicals, they steal electrons from proteins, lipids, DNA, and then those molecules will steal electrons from other molecules, and you have a cascade of reactions called the redox reactions. And these reactions will end up hurting the cell. We are primarily interested in vascular endothelial cells. And as I said, when this balance is tipped in favor of reactive oxygen species, then the cells become dysfunctional. And when they become dysfunctional, they may become more prone to apoptosis. They lose, because of the less bioavailable nitric oxide, the vessel will undergo vasoconstriction. They also express inflammatory markers like the pro-inflammatory receptors, where leukocytes will adhere to the endothelial cells on the vessel wall, so we have inflammation. We also lose the protective effect that the endothelial cells exert on the smooth muscle cells, so now we have uncontrollable vascular smooth muscle cell proliferation, hypertrophy or hyperplasia, and we also have endothelial cells that are now prothrombotic. So when you add all these together, that contributes to the development of a chronic vascular disease called atherosclerosis. And atherosclerosis is the precursor to cardiovascular disease. Cardiovascular disease, despite all the progress in medicine, cardiovascular disease, and by that I mean if we add all the heart attacks and strokes, continues to be the #1 killer in the developed world, including the United States. So it is important to understand endothelial cell dysfunction, because this will allow us to prevent the development or progression of atherosclerosis, and hence it will allow us to prevent the development of cardiovascular disease. So in the past, the AVML had important contributions, primarily by our lab is an in vitro lab, and we exert mechanical forces on the cells and examine the cell behavior. So most of the time, we were doing fluid shear stress studies. So in the past, we delineated the mechanisms by which fluid shear stress regulates thrombosis and thrombolysis. We provided the first evidence, and myself and a couple of other labs in the country, that shear stress initiates reactive oxygen species mediated intracellular signaling in endothelial cells, and investigated the subcellular sources for this ROS production. The subcellular sources at the time, we concentrated on NADPH oxidase, which is a plasma membrane bound enzyme that is known to produce ROS under stress conditions. Later on, we switched to mitochondrial research. We delineated the intracellular signaling by which ischemia leads to endothelial cell injury, and the intracellular signaling by which these reactive oxygen species make the post-hypoxic endothelial cells pro-inflammatory. We also provided the first evidence that mitochondrial ROS regulates CR-induced gene expression, and by that I mean we examined the gene expression for antioxidant genes And we found out that when you have an overproduction of mitochondrial ROS, you actually, the endothelial cells may actually upregulate antioxidant gene expression. We also delineated the intracellular mechanism by which ischemia reperfusion, where for the first time we introduced flow as a component of reperfusion. So basically, besides doing the traditional hypoxia reoxygenation static experiments, we simulated ischemia, and then upon reperfusion, simultaneously we introduced reoxygenation and flow, and we found and we quantified mitochondria and endothelial cell dysfunction. We also studied the role of bioenergetics in mitochondrial dynamics, a very hot field because the mitochondria are undergoing continuous fusion and fission, and we studied this and quantified mitochondrial dynamics and motility. In the last few years, we, after especially the move to University of Buffalo, we demonstrated the role of mitochondrial calcium uptake. We had a PhD student, I'm going to show a slide from his published data. We concentrated on this channel that is called the mitochondrial calcium uniporter complex or channel. This is the channel that controls calcium influx from the cytosol into the mitochondria, and that's the only way by which calcium can enter the mitochondria. So it's a very important complex. And for the first time, we showed that by controlling the mitochondrial calcium uptake, this regulates the shear-induced cytosolic calcium signals, of course, mitochondrial calcium signals, and cytosolic calcium signals, and hence it can control the fate of the cell. We showed also that if you apply chronically shear stress, you actually differentially modulate gene and protein expression of the complex subunits, and hence you can affect the MCU complex activity, which again controls how much calcium will come into the mitochondria, and this, the amount of calcium in the mitochondria may regulate the concentration of free radicals that is produced by the mitochondria, hence the concentration of reactive oxygen species in the cytosol. Hence, you have all the redox reactions. You may affect transcription, translation, the fate of the cell. So these are the level of cellular molecular activities that we have in the lab. And this is part of the work from the previous PhD student who is now working in North Carolina in a pharmaceutical company. And we showed, in a few words, we delineated the mitochondrial calcium signaling pathway in endothelial cells exposed to shear stress. And what we did differently is, well, for the first time, basically, somebody asked if we were able to measure intramitochondrial calcium signal in live endothelial cells that were subjected to shear stress. And we also were able to overexpress the critical subunit of the MCU. And we noticed that when MCU activity is increased, which other labs have shown that the activity of the MCU is increased under pathological conditions. For example, in diabetes, they believe that MCU is overactive and lets more calcium into the mitochondria. We showed that under shear stress, when you have increased mitochondrial calcium influx, these, of course, the increased calcium makes the electron transport chain produce more mitochondrial ROS. So you have the overproduction of mitochondrial ROS. And these mitochondrial ROS, together with the high intramitochondrial calcium, activate the mitochondrial permeability transition pore. This channel here, it's a low conductance, MPTP. It opens, allows ROS to go out of the mitochondria. The mitochondria are located very close to the endoplasmic reticulum. As soon as they exit, they actually oxidize the IP3 receptor, and then the IP3 receptor opens and allows calcium to spill out of the endoplasmic reticulum. Then, of course, since the mitochondria is right there next to the ER, enters through the VDAC, which is a channel that is always open to calcium. Then, of course, again enters through the MCU back into the mitochondria. And this circle, 1, 2, 3, 4, that keeps on repeating itself under shear stress, causes transient oscillations, basically transient increases in mitochondria mitochondrial ROS and in intramitochondrial calcium. So these oscillations, the frequency oscillations we quantified, and we believe that these are signals that are important for the fate of the vascular endothelium. So in summary, we showed that when you have increased endothelial cell MCO activity, this disrupts the normal calcium signaling in response to shear stress by causing these mitochondrial calcium oscillations, which may predispose the endothelium to mitochondrial dysfunction and cell dysfunction. And of course, we also showed that the calcium is replenished from the extracellular media through the piezo1 channel, that it helps replenish the calcium in the ER through the SERCA, and also the enzyme endothelial nitric oxide synthase is activated by calcium, releases nitric oxide, that has also contributes so that the electron transport chain will enhance its production of mitochondrial ROS. So now I'm going to move to the current research in the laboratory. We have projects. I'm going to talk to the one that I think, I'm going to talk to a couple of them, but mostly I'm going to talk about the first one, which is the role of endothelial cell-derived mitochondria carrying extracellular vesicles in hypertensive stretch-induced endothelial cell dysfunction. And I'm going to give some ideas about how to connect to an in vivo model via a collaborator at the University of Buffalo. So some of you, depending on which lab you are, you may already have heard about extracellular vesicles. 15 years ago, up to 15 years ago, I would say, if we were on a committee and we were looking at grants, colleagues of mine were very critical of extracellular vesicles. They would think that it's debris. And those grants are starting appearing saying that extracellular vesicles, which are vesicles released by the cells, both under physiological and pathological conditions, have a purpose. And the purpose today we know is intercellular communication. These vesicles carry proteins, lipids, DNA, and they can affect the function of other cells. And so today there are conferences. My MD PhD student is actually going next week to Malta to a small conference devoted solely on extracellular vesicles. So tremendous focus right now on extracellular vesicles, which, as I said, just the last decade all this happened. So how did this start? It started because they noticed that when they inject adult stem cells or other stem cells, we have paracrine effects in cardiovascular disease. And basically these stem cells, initially everybody thought that they hone on the host and differentiate to different cell types. But then eventually we realized that it's soluble factors that they release, the cells release. And that's why the stem cells had paracrine effects. And by paracrine effects, what do I mean? Well, they were protecting the myocardium, so they had they enhanced some cardiac metabolism, they caused cardiac regeneration, neovascularization, cardiac remodeling. Basically, they have beneficial effects to cardiomyocytes, endothelial cells, smooth muscle cells, fibroblasts, and cardiac stem cells. So in 2008, there was a study where they took the conditioned media from these stem cells and they split them into two portions. One where they concentrated all the particles in the conditioned media that had less than 100 nanometers diameter, and they had less than 1,000 kilodalton molecular weight, and one where they were the larger ones that were between 100 nanometers and 1 micron, and they were heavier than 1,000 kilodaltons. And they noticed that when they incubated with to see the paracrine effects, when they incubated this portion of the particles with the cells, they noticed that this portion was the one that had the cytoprotective effects. And today we call them the large extracellular vesicles, because the other ones were the small extracellular vesicles. And talking about small versus large, even up to now, every couple of years there are guidelines how to name, how to characterize the extracellular vesicles, and still up to now, tremendous lack of consensus what to call them, how to isolate them from the conditioned media. So there is no established consensus regarding extracellular vesicle nomenclature according to the latest guidelines, which was last year. But what we know is that every cell releases 3 types of extracellular vesicles. The small ones, size 50 to 200 nanometers, they are also called exosomes. They are packaged via the endosomal sorting machinery. They are released within MVBs, which are multivesicular bodies, which are basically late endosomes. And then they also release what we call now the large, sometimes they're called medium to large vesicles, and they also call them microvesicles or microparticles. And the head size, I would say between 100 and 1,000, sometimes between 100 and 1,500 nanometers. They are released by blebbing of the plasma membrane during physiological conditions and also pathological conditions. And last, the very large ones, they are called apoptotic bodies. They are released when the cells are dying, basically during apoptosis, again by plasma membrane blebbing. And of course, to release anything, to have the budding of the plasma membrane, you need cooperation with cytoskeletal elements. And more recently, they found that in these large extracellular vesicles, some studies found them also in the small extracellular vesicles, but they primarily, there is some agreement that it is mostly the large extracellular vesicles, that some of them contain mitochondria. And it kind of makes sense because the average size of a mitochondrion is 1 micron. So, you know, if you want to have a whole mitochondrion, then definitely you need to be in the large EV category. Of course, if you have mitochondria fragments, they can fit anywhere, large, medium, small. And you can see here in this EM picture, this extracellular vesicle contains mitochondria. Are they whole or fragments? We can speculate. So we call these EVs that carry mitochondria cargo mito-EVs. And today we know that they are released by every cell type, endothelial cells, neutrophils, macrophages, epithelial cells, neuronal cells, etc. And recent reports suggest that the large EVs primarily are responsible for transferring mitochondria to other cell types. And same cell types further downstream, let's say, neighboring cells. So the mitochondria that are transferred, several labs have shown that they can integrate into the recipient cell's mitochondrial network. These are the target cells. They transfer themselves, they reach the target cells, or you incubate them if you are in vitro lab, and they actually can go inside and they can incorporate themselves with the mitochondria of the target cell. Now, and the amazing thing is that labs reported that these mito-EVs can elicit restorative effects in the recipient cells. So for example, for endothelial cells, they found that brain endothelial cell-derived EVs actually increase the ATP levels, the ATP production, and improve the viability in endothelial cells that were injured, damaged, by exposing them to hypoxia. So if you have hypoxic endothelial cells and then they incubated and EVs from healthy cells, then they see an improvement in the function of the hypoxic endothelial cells. At the same time, other labs, these are recent studies, 2021, 2019, showed that mito-EVs can actually cause harm to the recipient cells, can induce cytotoxic signaling in recipient cells. In this case, they got, in this study, they got EVs from endothelial cells treated with lipopolysaccharide, and these were EVs that, again, some of them, a portion of them, carry mitochondria, and when they incubated them with the recipient cells that were healthy, they noticed that they made the healthy target cells pro-inflammatory. So actually, you have this dual role where they can do good, but they can do harm. And now in recent papers, 2021, up to 2024, you're starting to see more knowledge about microvesicles, microparticles, which are these large EVs. They've been endocytosed with different, or pinocytosed, different methods to the recipient cells, and they can cause many, many effects. And basically, here in the schematic, you see here is a donor cell, it buds off the EVs. Some of them contain mitochondria. They are, in essence, taken up by other cells. And what do they do to the recipient cells? How do they do it? How can we regulate that? This is of interest to us. And so we have this dual mito-EV paracrine model. This is a sketch that Zahid Mazar from my current PhD student put together, and he is the one who single-handedly started the EV project in the lab, came to the lab and said, I want to study EVs. And I was like, but there are too many, they are too small, I don't know how to help you. But he took it from zero and brought it to the stage where I'll show you the results today. But basically, we believe, and people believe, that when you have mito-EVs from stress cells, and then you expose healthy cells to these mito-EVs, they become inflamed or dysfunctional. And when you have EVs from healthy cells and you expose them to cells that have been damaged, they may increase the ATP production, decrease the ROS production, cause other beneficial effects. I'm not sure what Zahid put on the slide, but anyway, they can make dysfunctional cells back, restore some of the health of the cells. And the application of that, or at least one of the applications we chose, was in collaboration with the Way Lab at the University of Buffalo. They have a pig model of repetitive pressure overload. They call it RPO. This is where they expose the pig circulation to phenylephrine, so they increase the blood pressure in the pig only 1 hour per day. So it's a transient hypertension similar to what adults would face who are hypertensive and don't control the blood pressure well. So it's only 1 hour a day of hypertension. And they noticed that after 14 days of having only 1 hour per day of hypertension, they noticed that there was some left ventricular diastolic dysfunction. They measured other parameters. I will show you some of their data in a later slide. And basically, they believe that just 1 hour hypertension per day can lead to heart failure. They call it heart failure with preserved ejection fraction. And I would call it a HFpEF-like cardiac phenotype, because obviously it doesn't. The human heart failure with preserved ejection fraction is such a complicated story. And I think this is one of the cons in any animal model. But anyway, we formed the hypothesis that due to the hypertensive stretch, the endothelial cells, the arterial endothelial cells, are releasing EVs, large EVs, that maybe they contain a dysfunctional mitochondrial cargo. And as these particles flow farther downstream, they reach, let's say, the coronary arterioles. They are absorbed by the coronary arterioles, and they form microvascular, dysfunctional microvascular endothelial cells. And we speculated that this would cause decreased oxidative phosphorylation, increased mitochondrial ROS and intracellular ROS, impaired nitric oxide signaling, increased inflammation. So, and this, of course, coronary microvascular endothelial cell dysfunction will lead to pathogenesis of heart failure. So that was the hypothesis. Easy to say, difficult to prove. When I think because I didn't make the slides large, it actually distorts a little bit. But you guys can I'll go over the text, whatever has been distorted. So Zahid, we decided, because the aim was so complex, to cut it down to a more realistic first hypothesis, testable hypothesis. So the hypothesis was that the quality of the mitochondrial cargo in these endothelial cell-derived EVs, in part, determines the paracrine effects on recipient endothelial cells. And the aim was to first identify, do endothelial cells release mito-EVs, and whether and how can we measure the polarization state of the mitochondrial cargo. Do these mito-EVs, if they have low polarization, When we incubate them with naive recipient endothelial cells, do they make them dysfunctional via donation of the mitochondrial cargo? And also, how can you devise strategies to improve the quality of the mitochondrial cargo and see whether these strategies will also abrogate the dysfunctional paracrine capacity of the mito-EVs? So Rita developed a protocol where they conditioned media from human umbilical vein endothelial cells following some sequential centrifugations. The last one is 20,000 Gs, and then takes the pellets, and this is the endothelial cell large EVs. And also we have 4 conditions. One is control cells in regular culture media, the cells that control cells that have been incubated 24 hours with mitotempo, which is a mitochondrial scavenging, reactive oxygen species scavenging a mitochondrial targeted antioxidant. And then endothelial cells that have been treated for 24 hours with TNF, tumor necrosis factor alpha, 10 nanograms per ml. This is kind of your positive control if you want to enhance the production of EVs. And then TNF treated at the same time with a mitotempo treated in the media. So we have these 4 conditions. And put here, we also pre-incubate half an hour with mitotempo and then both mitotempo and TNF. So we had to devise a way to characterize our EVs. EVs are known to externalize phosphatidylserine, and it is known that annexin 5 binds to phosphatidylserine in a calcium-dependent manner. So one way to visualize with flow cytometry your EVs is to stain them with annexin 5 Pascal. And then we use 2 other compounds. One is mitotracted green that recognizes any EV that has a mitochondrial cargo. And the other one is mitotracted red that is supposed to be sensitive to the polarization state of the mitochondrial cargo. So whatever EV, so all the EVs that have mitochondria will stain with annexin and mitotracted green, but only the ones that have functional respiring mitochondria will stain annexin 5, mitotracted green, and mitotracted red. So, and then we do analysis, we measure the size distribution of the EVs through nanoparticle tracking analysis. We go to an imaging flow cytometer. We are fortunate that in drivable distance, we have the Roswell Cancer Center, so they have an imaging flow cytometer because these particles are too small for a regular flow cytometer, and luckily Zahi doesn't mind coming back and forth all the time. And then we also characterize the protein concentration of the EVs. That's an easy assay, a protein assay, and also doing the lab PCR, and if we want to do proteins, we do electrophoresis. So what we found, so this is data from the flow cytometry, and you can see the particles are annexin 5 positive because remember the annexin 5 binds to phosphatidylserine, and this binding is calcium dependent. So when you incubate with EDTA, you lose the signal. And also remember the EVs are made out of plasma membrane, so if you incubate them with Triton X100, they pop up, they burst. So again, you lose the signal. And also notice that all the particles are smaller than the 1 micron beads. So obviously the centrifugation and isolation work well. And then when we do the size distribution, we see that we are always between 100 and 1,000 microns. So according to the literature, these are they used to be called medium large, now they're called large. And also when we plot mitotracted green versus annexin 5, we see that we have 2 populations, distinct populations. A population that is mitotracted green positive and annexin 5 positive. So these are the EVs that have contained mitochondria, and we call them mito-EVs, and others started calling them mito-EVs. And they are usually about 50%, maybe 60%, and these are control endothelial cells. And then the rest of them, we call them depleted DEVs. It's depleted from mitochondrial cargo EVs, and they are again about 50%, maybe a little bit less, maybe a little bit more. And how do we know where to do the split? Is because we used RBC, red blood cell derived EVs, red blood cells do not contain mitochondria, and as a result, the EVs do not contain mitochondria, so that's a good negative control. So the beauty of the imaging flow cytometer is that you can actually look at every particle under fluorescence. And amazingly, I mean, you can recognize the particles with a bright field. And here you see the annexin 5. Here we just chose, and of course we checked many DEVs and mito-EVs, and you can see the stain of annexin 5. And then when you look for mitotractate green, you see only the mito-EVs have mitotractate green because of the mitochondrial cargo. And then when you overlap them, the red and the green, you see nicely that the mitotractate green is inside the annexin 5 because the membrane is outside, it contains the mitochondria inside. And the imaging flow cytometer allows you also to see in bright field or measure the size of the particles. And we saw also that the mito-EVs are on average significantly larger than the DEVs, which kind of makes sense because they need to carry the mitochondria. And to make sure that indeed the EVs carry mitochondria cargo even more, Zaki did some PCR against a nuclear gene and against 3 mitochondrial genes. And basically the way these experiments are done is you normalize to whatever level of the genes are expressed, and then you add DNAs that chews up all the extra membrane, the external, whatever nucleic acids are stuck to the membrane externally, so you chew these up, so then whatever you measure shows you only gene expression of the content of the particles, of the EVs. And you can see that, so we are looking only at the DNAs plus, positive, the ones that were treated with DNAs. And you can see that the particles express very little to non-nuclear genes. They shouldn't have any nuclear genes because they bud out of the plasma membrane, they contain a part of the cytosol and maybe some mitochondria inside. But you can see that they are enriched in mitochondrial genes, which again verified that indeed at least a portion of our EVs contains mitochondria. Now, we developed an assay to be able to check the polarization state of the mitochondria inside the EVs. Well, I want to remind you that the mitochondria have 2 membranes, an inner membrane and an outer membrane. And on the inner membrane, we have what is called the electron transport chain. The electron transport chain is very important because it produces ATP, and ATP is the energy currency of the cells. So we have all these complexes that form the electron transport chain, and in order for the mitochondria to be alive, to be functional, to have functional respiration, we have a proton gradient that maintains the mitochondria membrane potential, the delta psi m, across the inner mitochondria membrane. And the way we assess the delta psi m is using this dye, the mitotracker red dye. But if we incubate the cells with an uncoupler, such as FCCP, what does FCCP do? It basically pokes holes in the inner mitochondria membrane and destroys this proton gradient. And by diminishing the proton gradient across the inner mitochondria membrane, it depolarizes the mitochondria. And so what Zaki did is he has control EVs, and then he keeps on adding increasing concentrations of FCCP, and notice that the mitotracker red intensity gradually declines, which shows that he can, in a nice dose-dependent manner, depolarize the mitochondria inside the EVs. And also you can see it here, mitotracker red versus mitotracker green. When you add the highest concentration of FCCP, you see that nothing changes from mitotracker green, but the mitotracker red declines, which is what you want to see. So we decided to check whether, if we treat, well, we decided to check what is the numbers, the mitochondria cargo inside the EVs and the polarization state of mitochondrial cargo inside the EVs from control cells and for cells that were put under stress due to incubation with TNF-alpha. And we did these experiments with and without the mitotempo in an effort to see if we change the redox state of the donor cells, how does this affect the quality of the cargo in the extracellular vesicles. And as you can see here, these are the number of EVs times 1,000 per certain, I believe is per 10 to the 4th endothelial cells per 24 hours. So when we measure the number of EVs, we see that control cells and with and without mitotempo, they produce a certain number of EVs. The number significantly increases if they are under stress due to TNF. In the presence of TNF and mitotempo, we have a significant decline in the number of EVs. The percent of mito-EVs doesn't change between TNF and TNF-MT, but again is significantly higher. Significantly higher than the control and the MT, control plus MT. And then we did the frequency histogram for the mitotracker green. And when you concentrate at the EVs that are larger they have more mitotracker green associated with them. You can see that actually when the cells are stressed, like the TNF EVs and the TNF-MT EVs, you can see that these EVs have carry a larger mitochondrial cargo. They have more mass of mitochondria associated with them. You can see here above a certain level, you see that the TNF EVs and the TNF-MT EVs have more mitotracker green intensity than the control EVs and control MT EVs. And this of course shows if somebody measures the mean fluorescence intensity of this frequency histogram, and you can see again that the mean fluorescence intensity of the mitotracker green is higher in TNF EVs and TNF-MT EVs. Using the mitotracker red, we are, as I mentioned, we are trying to understand the quality of the mitochondrial cargo. And you can see that control EVs are polarized. If you treat them with a FICCP, they depolarize. The TNF EVs are depolarized, but if you have TNF plus MT, you see they go back to being polarized. So you can see it also in the mean fluorescence intensity. And if we decide based on the FCCP, control EVs with FCCP, we decide the level of mitotracker red where 95% of the EVs have depolarized mitochondrial cargo, then we can quantify the percent of mito-EVs that carry polarized mitochondria. And in this case, we see, of course, that the TNF-EV, only 20% of the mitochondrial cargo are polarized. 20% of the mito-EVs that have polarized mitochondrial cargo, but it is restored if in the presence of TNF we have the mitotempo. So basically this tells us that controlling the mitochondrial redox state of the donor endothelial cells determines the mito-EV numbers, the quality of the mito-EV mitochondrial cargo, the numbers of polarized mito-EVs. The other thing that we did is we wanted to see when they go inside the recipient cells, where do they go? And we incubated 24 hours EVs. We did it both with EVs from dysfunctional cells or with EVs from control cells. They always go in, and always a percentage of them ends up in the mitochondrial network. You see here, we labeled the endothelial cell mitochondrial network. You see beautiful mitochondrial network in this control endothelial cell. The mitochondrial network is perinuclear. It's like a bunch of spaghetti. And we stained red using the cell-like mitochondria RFP Bachman viral transduction. And then we incubated them with EVs where we had stained the mitochondria with mitotracker green. And we can see that indeed there is colocalization of green and red in magnification here. And we did some Z sections. We can see that actually this green belongs to the mitochondrial network of the recipient cells. So it is possible after they go inside and incorporate themselves with mitochondrial network that they contribute to ATP production, they affect the mitochondrial ROS, as a result they affect the redox state of the cell, they can affect transcription and translation, they basically play an important role in determining the fate of the cells. So our goal is by modulating the quantity and quality of the EV mitochondrial cargo to then use these EVs to treat donor endothelial cells. We treat donor endothelial cells with a mitoprotective agent, and we believe that by modulating the quantity and quality of the EV mitochondrial cargo, we will be able to determine the EV paracrine effects. So here we use one pro-inflammatory gene, MCP1, monocyte motactic protein 1, and we see a very nice dose-dependent increase in TNF EVs from cells that were treated with TNF, and then we measure gene expression of MCP1 in the recipient cells, and we see that the more EVs we incubate with the cells, the more upregulation of MCP1 in the target cells, which is what we wanted to see. And then we took out of this, let's say, 15,000 TNF EVs per well, and we measured normalized expression of different pro-inflammatory genes in the recipient cells. So we did ICAM1, VCAM1, these are pro-inflammatory receptors on endothelial cells, MCP1, as I showed you here, and one of the interleukins. And indeed, most of the times we see, and these experiments are still ongoing, but most of the times we see that when you have control EVs or control EVs treated with with MT, these EVs do not induce inflammation to recipient cells. But when you incubate the recipient cells with TNF EVs, that's EVs from cells that were treated with TNF, or with TNF and T EVs, that's EVs from cells that were treated with TNF plus MT, then you see interesting phenomenon. You see that the TNF EVs induce inflammation to the donor endothelial cells, but if these EVs are from TNF/MT treated donor cells, they do not induce inflammation, or they induce significantly less than the TNF EVs. And that was very interesting to us. And this is where we are currently. So we developed a protocol to characterize quantity and quality of the mitochondrial cargo in large EVs that are released from control and TNF-treated endothelial cells. We showed that large EVs from TNF-treated endothelial cells, but not control EVs, upregulated inflammatory genes in recipient endothelial cells in a dose-dependent manner. That these TNF EVs carried on average and larger and more depolarized mitochondrial cargo compared to control EVs. That when donor endothelial cells were treated with TNF in the presence of mitotempo, then these TNF/MT EVs had the mitochondrial cargo polarization restored to that of control EVs, and they also lost their capacity to cause pro-inflammatory paracrine effects, suggesting that the mitochondrial redox state of the donor endothelial cells, at least in part, determines the mitochondrial cargo quality and the subsequent paracrine effects in recipient endothelial cells. And upon incubation of either control EVs or TNF EVs with the recipient naive endothelial cells, it appears that the mitochondrial cargo transfers and colocalizes with the mitochondrial network of the recipient endothelial cells, suggesting that these transcriptional EV-mediated paracrine effects are probably due, or at least in part, are due to donation of the mitochondrial cargo, subsequent changes in the bioenergetics and redox state of the recipient endothelial cells. And hence, indeed, I agree today that the EVs have immense potential as biomarkers, delivery vehicles, and therapeutic targets in cardiovascular disease. And we, again, in effort to connect to the hypertensive-induced heart failure that my colleagues have at the University of Buffalo, my PhD student Julia Petromica and master student Rafi Rajan, we have a stretching machine where the endothelial cells are cultured on a compliant membrane, on a loading post, and then when we apply vacuum across the periphery, then we apply equiaxial stretch, and basically we embark into creating too large, in my opinion, variety of stretch regimes. One of them is not what we call normotensive cyclic stretch. We try to be close to 1 hertz, although in the literature people are using different frequencies. But anyway, the normotensive cyclic stretch is, the percent allocation is 5%, and then the hypertensive cyclic stretch, usually it's around 15%. Frequency the same. And also we did some transients, because remember the peak model of my colleague is only 1 hour per day of hypertension apparently causes a phenotype to the peak. So we embarked into different transient cyclic stretches, both in when to apply the 1 hour or 2 hours, over a 12-hour period, over a 24-hour period. Initially we start usually with normal tension, and at some point we introduce the hypertension, then we go back to normal tension, and of course we control the extent, the percent allocation. Julia and Arun did 10% or 15%, and we call this the transient cyclic stress 10 or the transient cyclic stress 15. And we try to, we collect it, and these are preliminary data, but basically we see that the number of EVs increases when the endothelial cells release EVs. The number increases when the endothelial cells are subjected to transient 15 and continuous hypertensive cyclic stretch, and the number of micro-EVs, these are still ongoing, but interestingly, still we see that when the cells are hyperstretched, either transiently or continuously, we have a percentage of EVs with larger mitochondrial cargo, and also we see that they have disproportionately a high number of depolarized mitochondria in them when they're coming out of cells that are exposed to hypertensive stretch. So basically we think that since we believe that since these hyperstretch, hypertension-induced stretch causes, diminishes the quality of the mitochondrial cargo in the endothelial cell-derived EVs, these EVs can cause pro-inflammatory paracrine effects, and we believe that this might be part of the mechanism that causes endothelial cell dysfunction in the pig and maybe in humans too, due to hypertension, and may predispose to heart failure. So this is my attempt to explain my colleagues, Dr. Weil's swine model, and bear with me, whoever knows of animal models. The swine model of repeated pressure overload, as I showed this picture before, they introduce hypertension 1 hour per day, and what is really amazing, and the reviewers didn't like this aspect, that they can see heart failure in 2 weeks, or they think they see heart failure in 2 weeks. But basically 2 weeks of repeated pressure overload was found to significantly reduce the left ventricular diastolic compliance. So you see here, after 2 weeks of this regime treatment, you see that when you plot the left ventricular end diastolic pressure versus the left ventricular end diastolic volume, pressure per volume, you see that the curve shifted to the left and upward, and this shows that you have increased myocardial stiffening in the left ventricle. And also when they do histology, they see significantly increased fibrosis in the myocardium. And these are again signs that you have a heart failure phenotype, cardiac phenotype. The question is, do these pigs have endothelial mitochondrial coronary artery microvascular dysfunction? Do they have endothelial dysfunction? And the way this is done in a large animal model, is in order they test the vasodilation to a chemical called adenosine, and the way they measure myocardial blood flow is they inject microspheres that are fluorescent labeled with different colors. And post-mortem, they cut in pieces the myocardium and measure the density of the microspheres per gram of tissue. So you can see that at baseline, the myocardial blood flow is very good. Basically, you have increased myocardial blood flow in response to adenosine. But 1 hour after the 1 hour of hypertensive stretch, you lose the vasodilation. 24 hours after this one single episode of hypertension, the vasodilation is restored. However, if you have 3 days where you had in each day the 1 hour of hypertension, you do not get the vasodilation to adenosine. And of course, if you continued for 2 weeks, again, you lost the vasodilation to adenosine, which shows that as early as in 3 days of repetitive pressure overload, you do have endothelial microvascular dysfunction in the coronaries. And of course, you need 2 weeks to have this beginning of heart failure. So this basically tells my colleagues in cardiovascular medicine at University of Buffalo that the microvascular endothelial dysfunction precedes the left ventricular diastolic stiffening. And that's why we believe that endothelial cell dysfunction is important in this heart failure animal model. So our plans are to investigate the role of mitochondrial transfer by endothelial cell-derived microEVs in the context of hypertension and hypertension-induced heart failure, to assess the paracrine effects of the EVs released from cultured human endothelial cells exposed to different profiles of cyclic stretch, to get these EVs from blood also of the pigs, subjecting to different partial pressure overload profiles, and incubate them to target cultured endothelial cells from human and swine, respectively, to investigate the molecular mechanisms or cause by which the mitochondrial cargo in the EVs regulates target endothelial cell metabolism and signaling, to determine the translational potential of endothelial cell-derived EVs by isolating blood of the RPO-exposed swine, see if they cause microvascular dysfunction, inflammation, or think of some ex vivo in isolate arterioles from healthy swine, isolate them for blood of healthy swine, see if they can ameliorate endothelial cell dysfunction in coronary arterioles harvested from swine that have already become, they have heart failure. So all sorts of thoughts and a lot of work to be done. So I'm going to switch gears. I wouldn't be able to cover everything, but I'm just going to use a little bit more time to say something. Since I started with stretch and the whole talk was on stretch, I will say that we are also in collaboration with a colleague in chemistry who is a lipids expert. We also thought that deregulation of lipid metabolism might play a role in endothelial cell dysfunction under hypertensive stretch. And the way we did that, I will not go into the details, but the last few years there is an explosion of information regarding a particular category of lipids which are called sphingolipids. And we believe, in the literature it is believed that when you have an overproduction of ceramides, which are produced on the plasma membrane, in the lysosomes, and of course on the ER membrane, when you have an overproduction of ceramides, and in reality it's the ratio of ceramides to S1P, which is this molecule here, then this is an index of endothelial cell dysfunction. So we are after understanding whether the regulation of the sphingolipid metabolism also mediates in part and explains the dysfunction of endothelial cells that are exposed to hypertensive stretch. And we did some RT-qPCR to see, and of course when the cells are exposed to hypertensive stretch compared to normal tensive stretch, we see some upregulation of inflammatory genes. We also see some upregulation, small upregulation, but these are repeated now, of expression of the metabolic genes that are responsible for some of the reactions that generate the ceramides. So we see some upregulation of these genes, which may be indicative of overproduction of ceramides. And through my colleague, we hope to measure and quantify ceramides versus other products of the sphingolipid metabolism and understand more what is happening to endothelial cells under stretch. Because of the time, and because I already, this was very interesting, but anyway, another time, I will move to the acknowledgements. I want to thank the members of my lab. I highlighted the work of the ones that I presented today. All my current collaborators, local at University of Buffalo and long distance, and our agencies, especially the National Institute of Health, they also funded through a pre-doctoral MD PhD fellowship Zahid Manzar and the private foundations and some seed funds from the State University of New York. Thank you very much.
Thank you, Professor Alevriadou. A rich presentation. I don't know what to say other than it's an incredible, a lot of information. And I was getting some questions also while I was sitting here. So, but let's ask the audience on the EVs, which I enjoyed listening. Let's ask the audience, please.
I have two.
Can you speak up a little bit?
Yes, I have two questions. My first question is the purpose of stressing cells to induce apoptosis in the donor cells?
The dysfunction can be measured by different indices. So we measure usually with RT-qPCR to see upregulation of pro-inflammatory genes. But somebody can do proteins, and actually we're doing some Western blots to see if the proteins are upregulated, because upregulation of genes doesn't mean necessarily that the proteins will be upregulated. And then of course sometimes you do activity of enzymes, or so not necessarily, the cells do not become necessarily apoptotic, but when they are hit with an additional stimulus, then they develop more apoptosis. If, so for example, right now I didn't show this data, but when we expose the cells to hypertensive stress versus normotensive stress, and then we expose them maybe 2 hours to 4 hours TNF alpha, then those cells that have been exposed for 24 hours to hypertensive stress express more of pro-inflammatory proteins. So obviously the fact that they face hypertensive stress has an effect on them, but the effect may not show clearly unless you have an additional chemical stimulus. So it may make them prone to apoptosis under certain conditions. So not apoptosis per se, but in the presence of additional stimuli, they may more easily undergo apoptosis. Yes.
And my second question is, can mitochondrial transfer happen between cells?
What's the second one?
Oh, sorry.
Yes.
Can mitochondrial transfer happen between cells of different types? And if so, does that greatly affect paracrine effects or?
Yes.
Let's repeat the question so that we have, because people are listening to it also. So let's
Yes.
one more time.
Yes. The question is, a very good question, both of them. Thank you. The question is, will EVs that come out of one cell type, will they actually affect only same cells farther downstream, or will they be absorbed by other cell types? Part of the complication in this type of research is the problem that in vivo, all the cells release, and amazingly, different cell types absorb them. They endocytose them or pinocytose them. So it's very hard to pinpoint how important are, as an in vitro person, I know what my cells are doing, but it's very hard to project in the in vivo environment. However, there are labs, and it kind of makes sense that when you have EVs, let's say from endothelial cells, they came out of the plasma membrane of an endothelial cell, they have receptors to bind to other endothelial cells. So preferentially, there is going to be some endocytosis by endothelial cells farther downstream, especially for endothelial cells, since they are flowing with the blood. Does this mean that endothelial cells will not absorb EVs from other cell types? They will absorb everything, but there may be a preference to EVs that came out of endothelial cells upstream. So it is very complicated. There are also indications in vivos. For example, we know that hypertensives or people who have coronary artery disease, they have in the bloodstream increased numbers of endothelial cell-derived EVs. So we know that already. What does this mean in reality? Nobody knows. But so we have some human indications that this may be important. And then you have the in vitro experiments, and it's, as I said, hard to translate them to animals and humans. Very good questions. Thank you.
All right, we're running out of time, but maybe 5 more minutes. So
Yes,
you know, anybody has any questions further on that? Okay, I want to ask a question. You know that we are, Professor Coulter, Raj, and I are working on creating electronic studies techniques. So,
Now you've got it. Don't ask me anything difficult. Yeah.
and so his particular question says, if creating an organ on a chip, or organoids basically, right, emulate this activity, can we also create, when we study these cells, you know, particularly the EV properties, can we also create a hypertensive environment for stretch and fluid, you know, shear stresses as well? And I think you kind of answered that at this moment, that it seems to be challenging to do all of these things. But I'd rather have you address
Very
that.
challenging. So if you start trying to create an in vitro model that incorporates the complex mechanochemical environment that the cells encounter in vivo, it's very complicated. Very few labs started joining forces. Let's say you want to incorporate shear stress and stretch. It's very complicated. That's why most labs, the in vitro labs, isolate one type force. Like our shear stress experiments are very different from our stretch experiments because the equipment is different. And also to do molecular cellular work, a lot of times you need to have the possibility, the capability to put the cells under the microscope. This creates problems.
And also put them in, to emulate the environment of the body as well, which is another.
Very complicated. Yes.
Well, thank you very much. If we don't have any other questions, let's give a hand to Professor Alevriadou.
Rita, welcome. I just